Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, part of the Finding Genius Foundation. My guest today is Kristin Swanson. She's the Vasek and Anna Maria Polak Professor in Cancer Research. She's also co-director of the Precision Neurotherapeutics Innovation Program, a director of the Mathematical Neuro-Oncology Lab. All of this is part of Mayo Clinic in Arizona. Many designations and accolades that she has. So we'll just get into it. And Kristen, thanks for coming. How you doing?
I'm doing great. How are you?
Good. Well, tell me about your research. What's the focus of it?
So my lab is the Mathematical Neuro-Oncology Lab. And so it's everything that name entails, the idea of anything mathematical and quantitative applied to brain cancer patients. So we optimize patient care, leveraging quantitative tools, everything from mathematical modeling, like hurricane prediction for brain tumors, all the way to like artificial intelligence, machine learning sort of approaches.
Well, what are you trying to model about tumors? Like how they spread or when they'll metastasize or what?
Yeah. So the type of brain cancer we work on is our primary brain tumors known as gliomas, the most aggressive of which you might have heard of is a glioblastoma. Beau Biden had glioblastoma, for example. John McCain had glioblastoma. It's a very aggressive, diffusely invasive disease. And what that means is the cells spread beyond where you can see them on an MRI or on any other imaging or even really surgically. So the tumor cells kind of spread long distances. And so one aspect of what we do is build spatial and temporal predictive models of where each patient's tumor is spreading. So we can optimize things like radiation, where to dose radiation, and predict how or which treatments a patient will best respond to. And then the other aspect of tumor biology that we sort of focus on is the idea of intratumoral heterogeneity, the idea that you biopsy in different locations within a single tumor, you get lots of different biology at play, and drug or a treatment usually only targets one of those. We need to know what all is going on in the tumor in order to know how to tackle it effectively.
So what are you trying to model about the tumor? Are you trying to model the heterogeneity of the genetic underpinnings of the cells in it, or just literally 3D spatially what it looks like, or what do you model?
Yeah, so it's kind of two orthogonal things. On the one side, it's 3D spatial. So we build these 3D spatial models of temporal evolution. So it's literally a hurricane map for each patient's tumor. But that's kind of the overall abundance of tumor cells and where they're going and when they're going there. But the intratumoral heterogeneity aspect, we collect image localized biopsies. And what I mean by that is you have a patient and an MRI of their brain. And you then, when the patient comes to the surgical suite, when tissue is collected, when the tissue is removed from the brain, when the tumor is removed from the brain, you know where you're at anatomically on the MRI. So you could say, oh, well, when I send this little piece of tissue to pathology to look at it under the microscope and tell me what kind of disease it is, I also know where that's located relative to the MRI. So I can learn the patterns of what's on the MRI to predict what the biology is the pathologist reads. So like I could predict, one of the things that we predict and work on is looking at intratumoral heterogeneity of the molecular diversity of the disease. And what I mean that is gene A is broken here, but gene B is broken there, and being able to predict where gene A and gene B are broken. And the reason you'd want to do that is because tumors, like I said before, respond to treatments differently, right? Different portions of the tumor will respond differently. And if you have a drug that targets, say, brokenness of gene A, then you would only expect those tumor cells that are broken in gene A to be responsive, and the gene B guys need something else.
Well, what do you notice 3D spatially? What do tumors tend to look like? What's the distribution of heterogeneity? Does it follow a pattern in evolution?
Yeah, it depends on a lot of things. So it depends on the evolutionary biology of the mutation or the gene target you're looking at. But so let's just talk about clonal history. So you hear about tumors and having that they have clones or subclones, and that, if you could figure out what the base thing that caused this to be the primary clone, the base event, the base mutation that sprung out the next clone, then you could think of the primal clones, the primary clones, and then all the daughters from that progeny. And if you could get to the stem, you could basically nip it out in the bud as the bud. So if you could target that stem mutation is the idea, right? Then you can kind of kill all of the cells. In the case of GBM, it's very heterogeneous. There's a few of those sort of more stem-like mutations in this disease where you'll find if one tumor cell has it, a lot of the other cells have it. But then there's other aspects of this disease for which that's not true. There's a lot of other biology for which that's not true. So that's one of the key limiting factors of being able to really cure and make a significant impact in GBM. And GBM is probably one of the most aggressive tumors. GBM being glioblastoma is one of the most aggressive tumors there is, and one of the reasons for that aggressiveness is because of this heterogeneity within the tumor with regard to.
But again, what does that look like? Do tumors seem to grow from a center radial outwards, or are there multiple initiation points? And can you trace the lineage of certain mutations?
Yes, you can definitely trace the lineage of certain mutations, and it is definitely thought that there are areas of propensity for where tumors start, but it's not really radial, it's more anatomical. It's the cells migrate very quickly along what are called white matter fibers within the brain, so it's the long axon tails of neurons, and so you get this very asymmetric, non-spherical growth pattern of these tumors as a result of that. The tumor cells use these sort of highways to get further distances in some directions and shorter distances in other directions because the highways don't head that direction.
So is the tumor proliferating? I mean, so it's not necessarily one ball-like mass. It's pretty early on, it starts to move along these preferred highways, as I guess you mentioned it, and spread that way.
Correct. Yes, I kind of liken it to, I had one analogy at one point is, how you drop grains of sand onto a table and you can imagine them sort of individual sand granules rolling off of the pile. So visualize that as the cells, but then have each of those be able to move. So they're like cockroaches on top of that. So that's the kind of visual I analogize for what the actual behavior of these tumor cells are.
What appears to be are they attempting to make a structure, or based on how they move and spread and how the tumor grows? Again, it's not. Is it a monolithic thing, or is it clumps, or is it long filaments of cells, or what does it look like most closely?
The thing that makes it most unique is the fact that it's not really filaments. You think of things like breast cancer, right? They follow ducts and then they expand in certain directions, and then you'll get some expansion, but it's really mass-like in a lot of breast cancer, not all. But in brain cancer, it's not mass-like, although they have these individual cells have the opportunity of traversing along these white matter highways. They are sort of single cell, somewhat independent, but they're still a collective group, right? But they're not bound together or really as stuck together as you might see in other cancers. So they really have a cellular autonomous approach where each individual cell has its ability to do a lot of things on its own. It doesn't necessarily need to be pulling and tugging with neighboring cells. So the pattern is, it literally is those grains of sand. If you could remove all the brain tissue, you would see a pattern that's like dropping grains of sand onto a table with a pile in the middle of lots of them in the middle. But then single cell ones, some have rolled really far away and some are much closer.
Do you see that these cancer cells are attempting to recapitulate the structures of the brains from which they came? Or are they just, I don't know, they don't seem to have any particular function or structure or job, they're just expanding?
No, that's interesting. So there's a lot of things that's really fascinating about this disease. Their opportunity seems to be what they see as their opportunity, right? I think is that they want to build a community of things like them, right? And so they call on the normal responses within the brain to allow them to be there. So they incorporate sort of wound healing processes. So they peel back to those origins of saying there's wound, therefore incorporate all wound healing type cells to help facilitate tumor growth. So that's sort of on the immune axis. And then they also tap into sort of developmental pathways, like developmental biology, like how normal neurological development happens, to be able to expand and grow their community, right? That's what all cancers want to do, is they want to expand their population in a different controlled mechanism than what biology would allow for. The idea is that they've escaped the biological controls of growth. And in this case, invasion.
But again, are they performing any useful function, or is it completely self?
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Yeah, so that's a good question about how to think of it from a selfish perspective. They definitely produce certain biological products, but the thing that keeps coming up the way you ask that question in my mind is the fact that you can have a very significant tumor mass that you can see with the naked eye when you're in the surgical suite, yet that region of the brain can function nearly normally. So you can have normal motor function in a highly infiltrated brain region. So there is this facilitation where they can keep the behavior of the patient well managed, well able to be maintained despite having lots of tumor cells present. And one that could be a sign of the biology of the brain being able to adapt to insult, or the other side could be that this could be a strategy that the tumor is using to facilitate expansion.
Well, I just wonder if the tumor is trying to set up its own brain structure, or if it's just, again, capitalizing on what's there and it's not really doing any useful function. It's just cannibalizing where it can and taking over, but it doesn't seem to have any goal.
Yeah, I think the arguments for that is the fact that it does use cell-to-cell communication over long distances, right? So just like the brain does, right? So there is definitely some literature out of a couple different labs that are focused on the sort of synaptic function within synapses, meaning, signaling across neurons within brain tumor regions and that facilitating tumor growth, right? So the idea that there could be some synergism there, that they're trying to create communication networks, mini brain networks, right, across over long distances. There's some data to make sort of sense, if that's the direction you were thinking.
So in the brain, because the structures are somewhat diffuse, could you model it as primary tumor versus metastases? And, are there segregated, isolated lumps of tumor matter that, again, appear to use cell-to-cell communication? And if so, is there coordination amongst them? What kind of signaling is there, et cetera?
Yeah, no, it's a really good question. I think in this situation, that actually doesn't work, right? So there's just like a continuous gradient of tumor cells away from the center of the tumor mass, with the most being in the middle and the furthest, the least being far, far away. So back in the, many decades ago, in fact, to manage that aspect of this disease, they would perform entire lobectomies and hemispherectomies of patients, particularly young patients, pediatric patients with this disease, and find that the tumor would still recur on the contralateral side. So there's still tumor cells out there. So that going to your idea of the sort of long distance, or your question on long distance communication idea. But I was thinking about your question a minute ago about what's their strategy. There's some interesting stuff that we found in the perspective of biological sex differences underlying the tumor biology and the different strategies the tumors use in order to be tumors in male versus female brains, which might more directly speak to, I think, your question.
Yeah, what are the differences you see?
Yeah, so we did a biological study with this group, Josh Rubin's lab up at Washington University, and I think we published this last year, that was looking at sort of the more biology side, and then I'll tell you about the mathematical modeling side, or the imaging mathematical neuro-oncology side. So on the biological side, we basically took RNA-seq, so look at the molecular transcriptome of these patients, and looked at these cohorts, and we basically asked the question, what's common in the patterns of the molecular signatures of these tumors? And we subtracted what was common between male and female out. And then you look at the remainders, and the remainder has a female pile and a male pile. And if you just look at the differences between those two populations, and you specifically those two signatures, you just look at long-lived females, for example, patients with GBM that lived fairly long, the signature for those tumors, those patients that were associated especially with tumors that ultimately lived longer, had a signal for what are like integrin signaling. So that would suggest something like maybe invasion would be a strategy that these cells were using to be able to facilitate longer survival for the patient. And so just that's one thing to set aside.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
On the male side, the complete opposite was true. It was patients that lived long that were male. Their gene signatures were most related to cell cycle control, meaning the tumors weren't as proliferating as much. And so that makes a lot of sense, right? You'd expect a patient will live longer if their tumor isn't going as fast from a proliferation standpoint. And then maybe the alternative for the male, so that was for the male, and the alternative for the female was that there would be some sort of control of invasion that was facilitated for females to be able to create long-lived patients. So we took that information, we then scaled it up to the patient MRI level to look at the dynamics of the disease.
You mean the whole body?
Yeah, like the patient's brain MRI, because this disease really only stays in the brain. So it starts in the brain and stays in the brain. It very rarely metastasizes outside of the brain, although patients had like transplants of like livers. There are case reports of patients with liver transplant having GBM, ultimately being able to transmit GBM via that liver that they transplanted. So there is some evidence that the cells are circulating. It's just they're not leaving the brain in typically treated patients. It's extremely rare. So the interesting thing, so now you're looking at patient brain MRIs and you're just asking, okay, well, are there differences in these proliferation dynamics and invasion dynamics that we can infer from those images? And using the mathematical modeling techniques that we've built to be able to sort of predict the hurricane path for each of these tumors, we can estimate how invasive a given patient's tumor is and how proliferative a given patient's tumor is. And when we quantify that across hundreds of patients, we found that just like the molecular mechanisms suggested, if patients had a control of their invasion patterns, that mattered for women, but it didn't matter for men. So if their invasion patterns, their extent of tumor invasion was higher or lower, that had an impact on outcome for patients that were female, but not for male. And so that was sort of self-consistent with that biological mechanism story. And then further, female tumors tend to respond differently to treatment than males. In general, females live a few months longer than males do with this disease. And it's really only been the last few years that people have really looked at this biology that might underline that. They attributed it to lots of different sociological things, but not a lot of biological mechanisms. So one of the aspects we found there on the treatment response is that females respond better to the current standard of care therapy. So this whole idea of giving all the patients the same treatment when they have an unbelievably diversely heterogeneous disease, as I described before, and there's differences between the sexes on overlaying even that complexity of biology, it really begs the question of, where's the opportunity to look at more individualized treatment strategies and less of these pooled strategies, these standard of care strategies that are sort of suboptimal.
So this type of cancer does not metastasize anywhere outside of the brain? Like how far does it go in the most advanced cases?
It'll go down the spine. It'll go anywhere in the cerebral spinal fluid contained system. So the CNS, the central nervous system, it can extend throughout that. But generally speaking, it doesn't leave that. And, one argument is that the patient succumbs before the tumor would metastasize further. That's one theory, but it's not really fully known. It was so expected that it was such isolated to the brain itself, the CNS itself, the central nervous system itself, that patients were allowed to have, after postmortem have transplants. But that is.
Are there parts? I've heard that the cerebral spinal fluid, people don't know. Essentially, if you do like a material balance, I guess the brain creates it, but the sinks for it, where does it go in the body and end up? And if the brain's continuously making it, eventually the spine would explode if there's too much fluid. Where does it go? And can actually aggressive cancer, shed a light on, if the cancer gets there and you're like, how would it get there? Maybe that tells you where the CNS fluid goes.
Yeah, I think there's definitely several mechanisms. The CNS, the control system there is it's created within the brain, but there are outlets within the brain as well to circulate this and to divert this to the circulatory system, to outside of the CNS is what I'm trying to say. But I think the more likely mechanism, or more common mechanism, maybe not the only, but the more common mechanism in patients that have had that have metastasis outside of the CNS are probably patients for which they had a significant surgical intervention, and during the surgery the cells sort of escaped, and then they had sort of different exposures to where they could expand. That's one theory there. But I don't know a lot in that area, I think, because I don't think there's a lot to know in that area yet, or not a lot known in that area yet.
Well, again, when you look at the pictures of these tumors, you look at the morphology, and then you look at them on a molecular level, et cetera. What can you predict? Can you predict in which direction the tumor will go next, and where will it go? And what kind of things jump out at you when you look at this imagery and study this stuff? Yeah,
so we can absolutely predict which direction a tumor is going to go. We feel really confident, and we've developed lots of tools over the last 15 years to be able to do that. So we can basically take a patient's MRI and generate a predictive map of literally a hurricane map of where the disease is going from an imaging perspective. Where will I be able to see tumor on MRI in 3 months? What would that look like? And we have a very good system to be able to do that. That's one thing. Then in addition, so let's say you have some targeted treatment that targets, like I said, gene A is broken. So it targets brokenness of gene A. And since we have these image localized biopsies that allow us to read out where in the tumor the biology might be different, we can then learn the patterns on MRI that tell us, oh, well, that's where gene A is broken. So we can track that over time and then watch the regions that are vulnerable to treatment A, whatever the drug is, and watch those resolve, and then figure out what the other populations of tumor cells are in these patients. So because these tumor cells are still in the patient's brain, by definition of the fact that it's a patient's brain, and you're not going to be able to generate, you're not going to be able to remove all these diffusely invaded tumor cells. That's the primary problem in this disease. You have this complex disease inside an organ that you can't remove a lot of, right? To be able to collect tissue to figure out what's in the tumor. You, by definition, always leave tumor cells behind, and the heterogeneity of those tumor cells are relatively unknown. So how do you tackle that disease, right? And you're kind of hobbled in 15 different directions on how to manage that disease. So by building these imaging informed models, we can then overlay on the patient's MRI the vulnerabilities, like what treatment would work there but not there, and what treatment would work here but not there.
If you could see the lineages, how true are they? Do they always progress along similar lines, or do they diverge? And is it so heterogeneous as to be only somewhat predictive?
When you say lineage, do you mean direction, like physical direction?
No. Well, physical direction, but actually what's going to be, what kind of either epigenetic marks or mutations in the underlying genes will you observe? How predictive is that when you look at it?
Yes. No, I understand what you're saying now. So basically, given the clonal diversity of what the tumor sample is that you have under the microscope right now, what you have in your hand, not what's left in the patient, can you predict what's the next population is going to come up, and what next target? So if I kill off some major clone, the subclone populating.
Like if I label, I'm just making this up. The first mutation observed is number 1, and then 2 and 3 and 4. There's like 3 paths that this kind of brain tumor tends to take. I'm just making this up.
Yep.
And we get all the way out to like mutation 55. And you looked at, let's say, 1,000 brains, 1,000 of these tumors, and they all seem to follow along these 3 pathways. And now you get a new person, and you take out, part of the tumor, and you see they're up to like, all the 3 pathways are developed, and there are a certain, distance along the path. Can you then say, okay, this is probably what's going to happen in the progression, and where more tumors will show up in the near future? Is it that predictive?
Yeah, I wish that was true, and I'm going to say no, based on the data that we have right now. So currently, one, it's hard to capture the full molecular diversity of the disease because there's intrinsically always tumor cells far afield from where the surgical site is, always. So we have to guess a little bit about those tumor cells. But even when we have, let's say you're approaching a very deep tumor and you collect surgical biopsies along your way in, the molecular diversity of those tumor cells can still be quite high, right? And so even if you hit some sort of base mutation that's common to a lot of the tumor cells, let's say EGFR is a good one, you see that it's common to a lot of tumor cells. The further challenge with this disease is it's in the brain. And so getting drugs into the brain is its whole separate category of challenges, because the brain is evolutionarily preserved to not let bad things in. And generally these chemotherapies are thought from the brain perspective bad, right? So the blood-brain barrier impedes our ability to deliver those. So that optimal sort of test of being able to say, I know the clonal diversity of this piece of tissue that is in this patient's brain. And then therefore, if I just hit the base clone hard enough, then I can knock the whole thing out, or I can hit this clone. And I know that means this next clone will automatically pop up. The complexity of this disease from the drug delivery perspective overlaid on all the other complexity makes that practically impossible to be able to detect, unless the path that we've been going down is, like I was saying, using these image localized biopsies and then AI to build AI maps that converts a patient's routine MRI into a map of where's clone A, where's clone B, where's clone C, right? And when you do that, then you could think of like an adaptive therapy strategy, which I think is the direction you're trying to go, where you can target clone A and watch clone B get a little bit bigger. So then you add in a drug for clone B, and then, oh wait, no, clone C is going to show up now. And then you can cycle between the various therapies. The pragmatic limitation historically has been an inability to be able to see where those cells are. So that's why we've put so much effort over the last several years into these image localized biopsies, which are often collected like you have a deep tumor and you're going to collect tumor cells en route to the tumor, which you would have naturally removed during any surgery anyways, just these small biopsies you would have removed anyways during the surgery. So those cells would have already come out of the brain, but they wouldn't have been sent to pathology for quantification. They wouldn't have been sent to pathology because they weren't considered the most aggressive portion of the disease. You see what I mean?
Yeah,
so,
I mean, even looking at the parts that are, easily visible and taken out, is there that much heterogeneity that you still can't quantify it? Like how many, just on a ballpark, like how many differences are there in tumor cells? What is heterogeneity? Thousands, millions, dozens?
Yeah. Oh gosh, there's definitely a diverse heterogeneity, but there's probably 6 primary gene targets that are the most common gene mutation based things, right? The types of mutations for those particular genes can be widely heterogeneous, but there's about 6 primary sort of standard, most common targets you see across glioma. EGFR is one of them. EGFR amplification. PDGFR amplification is another. All of those stitched together are, it's 6 times the various versions in which you make those 6, right? To get EGFR amplification, you get it 30 different ways, right? And, there's X number of mutations. I can't even count. I wouldn't know, that can also manifest in terms of resulting in the phenotype of EGFR amplification, for example. So is
there a Pareto though? Like, if I imagine, I'm just gonna, again, making this up. Like if I imagine my lung, it starts out with a main bronchus and then branches and branches and branches, but still it has a pretty discernible structure, just like a tree. Is there the same phenomena with the heterogeneity of cancer? Like if you map it out, all the known mutations and pathways and lineages and all that, do you see any structure? Or is it just like, it looks like chaos?
I think what you're looking for is structure on the phenotype side, right? Because evolution doesn't select on the genotype side. It doesn't care what gene mechanisms you use to get to whatever the behavior is. It just cares about the behavior, the phenotype, right? So I think there's definitely a lot of structure, but the glue for that structure is on the phenotype side, not the genotype side. So it's not that EGFR is amplified. It's that what does the EGFR amplification mean in terms of the cellular strategies? Do you see what I'm saying? So I think
if you start with changes at the genetic level, I know phenotype can, a lot more factors goes into, creating phenotype. But if you back it up and look at the genetic modifications and changes, is that discernible? Does that have a structure? Does that tell you anything? Or it's not useful?
No, it tells you enough to tell you that there's probably 6 guys that are the main genes that matter. Okay. And that there are definitely one of the most interesting series of publications, not our work, was that there's, a sort of a limited number of identifiable clones based on current sort of approaches within a given tumor, based on specimens that may be limited, right? You may not be able to capture the full diversity of the disease because you're not capturing where all the tumor cells are, given that caveat. But that GBM thought to be maybe a relatively small. So the argument towards what you're trying to say, which is if you know what that structure is, then you can, have this small list of clones, clone A through clone D, and then you can just iterate on therapeutics that handle the competition or synergism or communalism or whatever the population dynamic is between each of these clones. And I think in an idealized situation, you might be able, the data suggests you might be able to get there. But I think the trick is that these, mutations are not really selected on, and therefore the treatments for them are not really selected on. They're not selected on, right? It's the phenotype, it's the behavior that results in, right? So like, I just think of like, my background coming from a, quantitative sciences perspective.
I mean, but they look like we have epigenetic marks, and that's in response to our phenotype interacting with our environment. And so it's essentially reporting back. It's not necessarily changing the underlying nucleotide sequences, but, it's methylating them, regulating genes. In the same way, cancer, is it like an organism that, again, has a phenotype, even though it's heterogeneous, and that's interacting with the environment, and then it's changing in response to that?
Yeah, no, I think that's a really cool way to think about the disease, right? Is like any disease, cancer in general. I mean, I think that's a really insightful way to think about it. What I was trying to say is that if you look up what are the common features of, let's say, a mutation, a common one that exists in this disease, it's always proliferation and invasion. Different ways for the tumor cells to get to their goal of proliferating or invading. And if that's the case, then it kind of the gene mechanisms by which they get there, the tumor cell population strategy doesn't seem to care, right? They don't care about which way they get there as long as they get there, right? And so if
that's what I mean. So they abandon, like the cells give up their old jobs of acting in the brain in the way they used to. And again, it doesn't seem apparent to you that they have any new jobs. Now all they do is they just want to proliferate and invade, and they've given up their old functionality. Is that what?
Well, sort of. Yeah, no, it's a good point. Sort of. I think it's more like the reason brain tumors are so horrific at their continual ability to continue to be bad actors is they tap into their basic developmental pathways, right? Of how did neurodevelopment happen that was evolved over, eons, right? And so that developmental pathways is such an essential function for the overall human program, like the beast, the animal, the human, in order the human function, if the development does not process for the central nervous system, then the cost is high for the overall, right? Whereas you can imagine other organs, that's less of a case, right? So that has to be a really perfected system. And so they're just reverting back so the opposite of kind of what you were saying is that they're reverting back, but they're reverting back to that system more than any others.
Do you still see, though, that all the cancer mass is acting as a unified whole with communication amongst the parts? Or do you think it's like every man for himself, meaning every cell for himself?
No, I definitely don't think it's every man or cell for himself. There's beautiful work out of Michelle Manji's group at Stanford and Frank Winkler's group out of Germany that basically speaks to this idea that there's a much more intrinsic communication dynamic and that it's much less cellular autonomous, right? There's the ability for the individual cells to wander off, but because the brain, if you look at the brain under a microscope, just like normal brain, the cells are connected with these tiny dendrite type like connections across long distances. And there's different categories of things like astrocytes in the brain that, have different scales associated with them over which distance they connect things, right? So the whole brain is about connectedness. So naturally the tumor is definitely has a similar connectedness. And in fact, there's some recent work out of the Winkler group that suggested that if you were to target and watch, a single cell die here, wherever here is, right, some distance away, the system will realize that there's a missing cell and any daughter cell that's produced over there will run to that spot to fill in. So going to that systems dynamics, I think there is definitely increasingly an appreciation for systems dynamics, but it's early days. There's only a handful of labs that I know of that are working in that space.
Yeah, early on you said, we can predict where the tumor is going to go next. Well, how do you know? What tells you that? Does the tumor make a certain shape? Like, I'm joking, but does it make like an arrow shape that points to the right? You know it's going to go there next. Like, how do you know where it's going to go?
It kind of goes back to those white matter highways I was telling you about earlier, right? So that if the brain has a certain anatomical structure, every brain is slightly different, but there's general rules that glue them all together. And those white matter highways are great facilitators for tumor invasion. It's very logical to see, that general direction is the logical next direction. The time course of that, number one, is what we build these mathematical models for, and the detailed morphologic heterogeneity of where that direction is where these models come in. These literally, it's like a hurricane map, right? It's no different than you're looking at a hurricane, you're going, hey, that's New Orleans right there. I bet it's going to hit towards New Orleans, right? It's sort of, you can see the trajectory that they're on.
But on a hurricane map, though, there'll be like high and low pressure fronts that are moving in directions. So what constitutes that in the cell world, in the cancer world? Like, what constitutes a gradient or a front that would take it in a direction?
So there's a couple different things. One is that the nutrient dependency within the brain is different, right? So gray matter has a much more nutrient-rich region than white matter, one can argue. It's highly cellular regions, but it's also nutrient-rich. So there are definitely heterogeneity in local regional, like, nutrient populations and where that sinks in sources, you can think of it that way, within the brain. So that's one aspect. But in general, they just want to flee and populate as much domain as they possibly can. So there are like I said, there's a couple of these highways, and that being the primary location of expansion. But it's kind of all directions are a go, from the strategy perspective. It's not quite as oriented. So it's somewhere between that whole sphere idea, it just going out in all directions, and overlay the morphology of the brain, add a little sprinkle of the fact that there's variation, some variation in the nutrient environment, and there you have it.
I mean, if there's upcoming resections, surgeries, can you instruct or ask, hey, while you're taking this stuff out, can you suck out a little bit of CNS from other parts of the brain and keep it as separate vials so we could analyze and see if there's tumor cells there distally? Then you wouldn't have to, I don't know if you can do this, but then you wouldn't have to like cut into the brain at a whole bunch of points and poke a bunch of holes. Maybe you could just swab some, or again, get a little bit of a couple drops of fluid from distal areas and see if there's upcoming problems or cells that have moved somewhere.
Yeah. So the way surgeries generally are done is they start at a single focus, which is at the center usually of what the mass is. Again, there's always tumor cells kind of in all directions out from that. So it's not that they go in multiple times. It's rare that they do. Surgically, in the case of GBM, what they would do for second or third surgeries would be a significant mass-like local recurrence or distant recurrence. They would return to the OR. But even in a given surgery, are you saying they're like, I guess because they're going to the brain, they're just making a hole that's barely big enough to get in there? Is it almost laparoscopic? There's a lot of interest in minimally invasive, or minimally invasive is not the right word because neurosurgery has a very specific meaning behind that. But small incision, not expand necessarily, if you can avoid it, not necessarily having a large craniotomy hole, is definitely something that's of interest for a variety of reasons.
Yeah,
but
that makes sense.
But what I was going to circle back to is just mention one bit is that these image localized biopsies kind of are doing exactly what you were asking, is like, can you biopsy where in those regions that you kind of you're going to injure anyway as you go to the surgery, right? They're going to be part of that. They're in the tumor region, but they're not the hotspot of what the surgeon is targeting. The surgeon targets what they can see on the MRI, but what they can see on the MRI only tells them a small fraction of is really the tip of the iceberg of the actual overall disease. And so when during the surgery, you're going to run into the other pieces of the iceberg, and so those image localized biopsies I was referring to earlier, those are what are collected. They're like other pieces of the iceberg that you wouldn't normally, that are not the center of the disease. They would normally have been removed. They would absolutely be part of the normal surgical removal, but they wouldn't have been the focus of what the neuropathologist looks at under the microscope, because it's hard to find those tumor cells. They know they're in there, but they're not going to easily see that under the microscope. So I'm going to go to the place that I know I can see them.
Okay. Yeah, that makes sense. I mean, would it be useful to do a spinal tap along with the surgery to to see way far away, what shows up there?
Yeah, there is definitely some interest in two things. One is circulating tumor DNA, which in the blood itself, and in the CNS. There is some work in the CNS. The relevance of the CNS is really when there's leptomeningeal spread, which is not a very common outcome of GBM, again, because it's kind of going from inside the brain parenchyma to begin kind of like on the surface of the brain, and there's a different biological process to that. So it's not the most common. So the CNS tells you a little bit, but the CNS is more useful in other brain cancers like lymphoma. CSF, I meant to say, sorry, the CSF.
Okay. Do you see any niche construction, feathering of the nest before the cancer spreads to certain areas?
Yeah, so we work closely with this group at Moffitt Cancer Center, specifically Sandy Anderson and Bob Gatenby, on things like that, right? The questions of like local regional heterogeneity and habitat formation within tumors. And there is absolutely that case. The tumor has an entire ecology around it, and different regions have different things flourishing and dying. And that's contributes to that molecular and just biologic heterogeneity of the disease.
Okay. And then have you looked to see if there's any microbiome in the brain? And if so, is there any local, like what's the microenvironment look like where there's tumor and where there's normal brain cells? And again, is there like a microbiome there that you see?
I don't know anything about the idea of microbiome as it relates to the brain. I do know that there's significant connections between neurobiology and neuroscience and the gut microbiome. So those would be the direct connections that are sort of the most direct connections that I simply know exist, but know nothing about.
No, I just didn't know if anyone has seen if there is a microbiome in the brain normally.
I truly don't know. It's a good question. It's a great question.
Okay. Yeah, I guess while they're in there, then maybe nudge them to see if you can do some like 16S sequencing on what's there, the brain juice that's near the tumor.
Right, exactly. And it's a good question.
Just a couple more questions. So clinically, what are your goals for the next year, 2, 3 years? Like what are you working on that's going to be helpful in your opinion?
So we've partnered. So this problem of this, exactly what you've been asking about, this heterogeneity of the disease and intratumoral heterogeneity of the disease, and how do you even track it and know if I push down on this clone, will another clone pop up, and being able to know which ones those are. We have built this now, growing suite of sort of more machine learning AI-based tools that convert a patient's MRI into maps of where those clones or tumor populations are. It could be clones, it could be where are the T cells in the brain, whatever it is, so that you can convert a patient's MRI into these maps of the biology at play across the entire brain. And as a result of those, we partnered with now, I think 9 or 10 clinical trials to deploy those sorts of maps relevant to each of those clinical trials to say, look, I've got a drug that, again, targets clone A. It would be great if, we could see that it's actually functioning in these patients. And what you find is that you could overlay those maps on the serial imaging of those patients and watch clone A decrease or not, and say, yes, the drug is functioning or not on that clone. But the real problem is this clone C over here. It just keeps showing up, and it's the real beast. And so we really need a combination therapy that's clone A, clone C, going to your point. Could you get sort of build these combinations and build a strategy to control this disease and ultimately eliminate this disease? So that's probably the next year is focused on a variety of those clinical trials. In addition, we are designing some new strategies for how to spatially and temporally optimize things like radiation therapy to match the fact that there's this differential invasion patterns for each of these tumors, and then tailor them in smart ways.
Are there variations that only show up after chemo or after radiation?
Variations meaning specific molecular events, or is that what you mean?
Yeah, like certain lineages that only arise after certain kinds of chemo or radiation.
No, there's a lot of data to suggest the clones are there beforehand, or the molecular, molecularly distinct populations are there beforehand. It's just that chemo or radiation, whatever the treatment is great at knocking out several of these. Some are popping up. Some new ones are popping up as a result of, the recurrence. In addition, things like radiation and chemotherapy, which is the, backbone of the standard of care for this disease, also induces a lot of mutagenesis, right? There's a ton of mutations as a result of things like temozolomide chemotherapy that's used in this disease. So you can have lots of populations with lots of new mutations brought into the game, and whether those are relevant to, and relevant and targetable is another question, but there's a lot more that's brought in as a function of these very cytotoxic treatments that are used.
Well, that's what I mean. I would think that chemo would cause the appearance of certain, mutations or lineages that weren't there before.
Absolutely
because it's
true.
a deliberate adaptation to. I mean, it seems like that's what cancer is. It's an ongoing adaptation to its environment, especially in light of pushback, from the immune system, et cetera. So I don't know. I would just think that, again, chemo and radiation, all that would cause certain things to arise that were not there.
Yeah, that's absolutely true. In the case of temozolomide chemotherapy, there's something called a hypermutation phenotype that results of exposure to lots of temozolomide, and temozolomide is the standard chemotherapy used disease. So it's fairly common to see these additional new mutations, whether those mutations have meaning like they're significantly changing the phenotype of cells. All of that is there's a diversity of work underway in that area.
Okay. Well, very good. What's the best way for people to find out more about your work? Where can they go?
mathmedicaloncology.org is my website.
Okay, that's pretty simple.
That's the easiest place to go find everything. So it's we usually keep things fairly updated.
Okay. Well, excellent. Yeah, Kristen, it's been a great call. And thank you so much for coming on the podcast. I appreciate it.
So nice to talk to you. Thank you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.